It's the season of changing seasons again. Have you caught a cold? In fact, everyone has various antibodies in their bodies that protect their health. Everyone inevitably wonders, what is an antibody? Today, ChinaPeptides Classroom will talk about antibodies. Antibody AB, also known as immunoglobulin, is a protein produced by the proliferation and differentiation of B cells into plasma cells after antigen stimulation. It can generate an immune response with foreign antigens, thereby resisting the invasion of pathogenic pathogens. The structure of antibodies. Composed of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains, connected by disulfide bonds. 1. Variable area. Also known as Zone V, the composition and arrangement of amino acids vary greatly. The variable regions of heavy and light chains are called VHVL. HVR, a region with higher changes in amino acid composition and arrangement sequence, containing 3 regions, namely HVR1, HVR2, and HVR3, also known as complementary determining regions, namely CDR1, CDR2, and CDR3, which play a key role in antigen recognition and binding. Skeleton region FR. The non-HVR part in the V region is divided into 4 parts, namely FR1, FR2, FR3, and FR4. 2. Constant region. Also known as the C region. The composition and arrangement order of the carbon terminal amino acids are relatively constant. The constant regions of heavy and light chains are called C, H, and Cl. 3. Hinge zone. Located between CH1 and CH2. Rich in proline. Easily degraded by proteases. Next, let's take a look at the complete structural diagram of the antibody. Classification of antibodies. Based on the differences in heavy chain antigen specificity, antibodies are classified into 5 categories, namely immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. There are 2 types of light chains, K chain and lambda chain. Auxiliary components of antibodies. J chain, a cysteine-rich peptide chain synthesized by plasma cells, connecting monomer Ab molecules into dimers or polymer. Secretory piece. An important component of dimers synthesized by mucosal epithelial cells. Main function. Mediate the transport of immunoglobulin A dimer from submucosa to mucosal surface. Protect dimers from being hydrolyzed by proteases on the mucosal surface. Hydrolyzed fragments of antibodies. Although intact antibodies are suitable for most immunoassay applications, the performance of certain steps requires the use of antibody fragments for enhancement. Papain hydrolysis fragments, antigen-binding fragments, and crystallizable fragments. Hydrolysis significance. Elucidate the molecular biological role of antibodies. Remove crystallizable fragments to reduce the occurrence of hypersensitivity reactions. That's all for today's introduction of antibody structure. If you want to continue learning in the future, you can follow the video of ChinaPeptides Classroom in the next issue. See you soon!